coming. So it's my honor to introduce our speaker, Professor Linda Partridge. I won't take a long time. In the interest of her talk, Professor Partridge will show her interest is centered on the genes and mechanisms that determine the rate of aging, and as well as their implications in health and disease. Dr. Partridge started her career at the University of Oxford, where she obtained an MA and a PhD. First, she studied the behavior and ecology of birds, and then after she started her own group at the University of Edinburgh, she then started working with flies, focusing more on the relationship between reproductive costs and aging. After some work on the evolution of the rate of aging, a breakthrough came, I would say, in the late 90s, in the beginnings of the 2000s, when Dr. Partridge and also other people in the field started to identify the genetic determinants of the lifespan in model organisms. I think probably it was a very exciting point in time, because it became really clear that pathways that are involved in nutrient sensing and growth regulation, I wouldn't expect that like at that time, I would say, notably including the insulin growth pathway and the TOR pathway, could directly affect the lifespan, and I think that she will talk a lot about this then. Since then, her work has been focused on dissecting how diet, metabolism, stress, and sex can affect our lifespan. Just as a little bit of history, Professor Partridge started her group at Edinburgh in 1976, where she became also professor of evolutionary biology. In 1994, she moved to UCL in London as the Weldon Professor in Biometry. And at UCL, she not only managed her group, but she was also the director of the Institute of Health Aging until last year. In parallel to that, since 2008, Dr. Partridge has been the founding director of the Max Planck Institute for Biology and Aging in Cologne, Germany. And she was the recipient of numerous awards. I will keep it really brief. She became a fellow of the Royal Society in 1996, was elected to the Academy of Medical Sciences in 2004, a former honorary member of the AAAS since 2010, and was appointed as a Dame Commander of the Order of the British Empire in 2009 for her service to science. So without further ado, I would ask you to join me in welcoming Dr. Partridge.
Thank you very much, Felipe, for a very kind introduction, and Anne for telling us about Ms. Saunders. So I'm going to talk about aging, and from a biological or evolutionary point of view, it's actually rather a peculiar trait, because it's defined as the decline in survival and fertility as the organism gets older. But of course, natural selection would tend to pull things in exactly the opposite direction, towards an immortal organism that reproduces copiously throughout its very long life. So there's really something to explain here. And I think it's actually a trait where it's rather easier to think about it and to understand it if we do think about how it evolves. Now, of course, one possible version of the aging process is that it's just inevitable. Organisms wear out like cars or washing machines, and there's nothing to explain. But actually, it's very clear that's not true. So the most obvious counterexample is germlines. They go back to the primeval soup. We all come from a single common ancestor. We all share approximately the same genetic code. So we know that, in principle, cell lineages can be practically immortal. And there are also organisms that don't age. These are just some of them. This is a sea anemone, this is hydra, and planarian worms. And what they have in common is that they're all essentially bags of stem cells, and they're very regenerative or can bud. And I think probably very importantly, they set aside their germlines very late in their lives. Almost any of these undifferentiated cells can contribute to the germline and the future of the species. So aging doesn't have to happen. And it's also not a secondary consequence of other things like body size. It's a trait that evolves in its own right. So these are just some extraordinary lifespans from nature. So as far as I know, the mammalian record holder is this bowhead whale at about 200 years. They were actually aged by the harpoon heads that were found embedded in them from the whaling era. This Greenland shark turned up about 5 years ago and was finally aged by the Smithsonian. And they can live for almost 400 years. And they don't even start breeding till they're 150. So wouldn't make a very good fishery, but a remarkable bit of biology. And also body size, you can see down here, really is not very relevant. So as far as I know, this is the animal record holder at 500 years. It's just a little bivalve mollusk that lives on the floor of the Atlantic. And these two mammals here both live extremely long considering their body size. So this is the naked mole rat, which is turning out to be very interesting in aging biology. It's intermediate in weight between a mouse and a rat, but it can live for 30 years. And for some reason, they seem to be free of cancer. They don't get tumours. So there's some very interesting, their genome's been sequenced now. There are a number of interesting features there, and one is a very peculiar extracellular matrix, which may explain part of the cancer biology. And then there's this bat. It's not a hibernating bat. It's active all the time. Weighs 8 grams. You can just see it sitting here on the tip of a finger, but it can live for almost 40 years in nature. These are natural lifespans, not in captivity. So it's clearly a trait that can evolve in its own right in different evolutionary lineages. So, sorry, I think we've got a loose connection here. We're okay, I'll keep away from it. So clearly there's something to explain here. We've got a disadvantageous trait. The differences between these species are genetic. Let me see if I can get this to project correctly again. Let me have a look. So we're at this slide, and we want to go present a view. Yeah, good. So there's really something to explain. And because it seemed such a peculiar situation, the early thinkers about it suggested that since it's clearly not of an individual advantage, it must be somehow good for groups, good for whole species or social groups. So various suggestions were that if an individual was old and decrepit, then it should get out of the way to create the reproductive opportunities for the young. Or perhaps aging had evolved because it shortened the generation time so that natural selection could act more quickly and more effectively. And as I'm sure many of you in this audience will recognize, those are examples of group selection. The idea that an individual allele might spread despite its disadvantage to the aging individual because it's good for the survival of a group. And it's very clear from many population genetic analyses that group selection is a really weak force compared with selection on individuals to produce progeny. And I actually think there are no clear examples of a case where aging has evolved because it's good for groups. And if anything, actually what's seen is the opposite, because there are a few examples in nature of post-reproductive survival, particularly in females, that turn out to be of advantage for the group to which those females, post-reproductive females, belong. And one particularly nice example came out last year. This is work on killer whales. So killer whales live in mixed-sex pods, and they have long lifespans. They can live for decades. And what we've got here is just some work looking at what happens to a killer whale if its grandmother dies. So what we have in red are whales whose grandmother has not died in the previous 2 years. Here, in blue, is what happens if the reproductive grandmother dies, and here is what happens in green if the post-reproductive grandmother dies. And these are for whales of different ages who've lost their grandmother within the last 2 years. And what you can see is that even when the whale has reached 20 years of age, it's a real survival disadvantage if you lose your post-reproductive grandmother. Presumably the difference here is that the reproductive grandmother is to some extent still competing with others in the pod for resources because she's reproductive. But here, what's being lost entirely, it turns out, is her knowledge. So in times of food shortage, these old females know good places for fishing, and they lead the pod to places where they can find food. So I think it's an absolutely clear example where post-reproductive survival is definitely of advantage to relatives within the group. So I think this idea that aging has evolved by group selection is out of the window now. There are no clear examples of it. So how does it evolve? Actually, I think it was worked out really some time ago now by two very clever people at UCL. Peter Medawar is much better known for his discovery of the mechanism of immune tolerance, self-tolerance, but he had a sideline interest in the evolution of aging, and Haldane, very well-known evolutionary geneticist. And what Peter Medawar pointed out, it seems obvious now, but it turned out to be key to it, which is that even if you've got an immortal organism, it's going to die because of the impact of disease, predation, and accident. So external hazard will mean that as a cohort of animals goes through its life, its numbers are going to decline in accordance with the schedule of hazard at different ages. So if we've got some sort of survival curve, I've drawn an arbitrary one here, what happens is that if an individual dies before it reaches the age of first reproduction, it loses everything. So selection is high and constant on survival until the animal starts reproducing. But then Haldane chipped in here and said, well, suppose there are mutations that have a specific age of onset. He was thinking particularly about the Huntington's mutation. So the average age of onset of symptoms with Huntington's disease is about 35. But over our evolutionary past, very few individuals live past the age of 40. So this would be the schedule of external hazard for humans with 40 about here. The Huntington's mutation's effects only become apparent about here. And what that means is that all the carriers of the Huntington's mutations who die of external hazard up to this time are going to do so at a rate no different from non-carriers of the mutation. Selection can't do anything about it because it can't see it. And only at this point, when you start to see the phenotype of the Huntington's mutation, can selection act. So it's only acting on this tiny proportion of survivors that are present at the age of onset. And what Haldane said was, well, perhaps that's what aging is. It's just the accumulative effects of nasty mutations that, whose phenotypic effects become apparent at different ages. That's called the mutation accumulation theory of aging. There's another twiddle on that basic idea, which was produced by George Williams. He's a very famous American evolutionary biologist. And what he pointed out was that there could be a class of mutations that are beneficial to the young. So during young adulthood, they increase survival, they increase competitive ability and reproductive success. Something is making the young that have them more successful. But this is happening at the cost of a bad effect at later ages. Something's going wrong later in life. And of course, with those mutations, they can get into the population by natural selection because it's going to act more strongly on this early beneficial effect than on this late deleterious effect. That might seem like a very odd kind of gene action to suggest, but actually it looks as though it's very important in the evolution of aging. So as Felipe mentioned, I have an evolutionary past, and one of the main ideas here is that there's a cost of reproduction. If an animal goes in for copious early reproduction, it may be pulling back on immune defense, somatic maintenance, and other things, so it's in worse conditions later on. And there's quite a bit of experimental evidence that that's important. But there are other effects as well, and we'll come to a couple of them later on. There are a number of reasons why you might see this kind of gene action, and it's probably very important in the evolution of aging. So I think what the evolutionary analysis of the trait tells us is that we're expecting the rate of aging to evolve in accordance with the level of extrinsic hazard, and that's broadly true. So things that can fly, that live up trees, that are poisonous, that are well-armed, tend to have longer intrinsic lifespans than ones that are less well defended. And it's evolving as a side effect rather than of selection in its favor, either of mutation pressure, so these late-acting mutations can reach a higher frequency under mutation selection balance. And so it's a side effect of mutation pressure, or of beneficial effects in youth. And very importantly, it's not a program process. It's not like development. There isn't a well-oiled machinery of genes switching on and off in different places, making sure that the right things happen at the right time and in the right tissue. It's a much more haphazard process than that. And that may explain partly why it's so variable between different individuals. So the phenotypes of aging vary quite a lot. And of course, humans and captive animals are now living to much greater ages than in their evolutionary past. And the later part of our lives and their lives has not been moulded by natural selection, and that's why we see things going wrong. So, talking about living to much longer than our evolutionary past, what is actually happening to human lifespans, or what has happened over the last few decades? This is a set of projected life expectancies in birth projected forward into the future at 2030 for humans in these countries for which there's good data. So what they've done is to take the age-specific survival rates now and accumulate them into a life history to project what will happen in the future. We've got women on the left and men on the right here. And very importantly in this paper, there's a projected increase for all of these populations. Human lifespan is going to continue to go up. Although the extent of the increase is quite variable, it's fastest in middle-income countries at the moment. It's slowing down a bit in wealthy countries. And you can see that by then, despite the current situation there, that South Korea is projected to be the world leader at the time. The UK is about halfway up for both sexes. But interestingly, there's not a very strong correlation between health and wealth. The USA is actually quite well down the pile. And you can see the sex difference here. So the x-axes for men and women are different here. In all of these countries, women outlive men, and they're projected to continue to do so. But unfortunately, they're not healthy extra years. Women have a much longer period of disability and illness at the end of life than men do. So just looking a little bit at the variation, you can see these are very fuzzy estimates even within these countries. There are also quite big differences in circumstances between different countries. So this is just what's been happening since 1960 in these high-income countries for which there's good data. This paper came out in Lancet last year. And you can see the steady increase. So this has actually been going on at about 2.5 years per decade since the middle of the 19th century. It is an absolutely amazing increase. But you can see the elevation of the lines very different for different countries, even without huge differences, distances between them. So England and Wales here are about average for the trend for men in these different countries, but Scotland is way down the pile. There's a huge difference in life expectancy between the two parts of mainland Britain. And if you look at women in the UK here, you can see that they're gradually sagging off the trend. And it's not clear at the moment why there's this sex difference. But clearly, within the overall trend, there's quite big variation going on. I mean, one idea about this effect in women is that it may be a past effect of women taking up smoking in the 50s and 60s. But whatever it is, you can see that the overall picture masks quite subtle differences between different places and even between the sexes within one. But overall, the thing is increasing. But what's very clear is that health span isn't keeping up with lifespan. So it's a little difficult to really compare historical data because they were collected in slightly different ways at different times. But really, however you cut it, there's a growing period of ill health at the end of life as lifespan increases. And that's why we're seeing this, the very strong relationship between age-related diseases and advancing age. So this is dementia, cardiovascular disease, and cancer, the three big ones. These are onset or prevalence data for either the UK or for the EU countries. And you can see this very strong increase with age. Dementia more prevalent in women than in men, the other two the other way around. And what's happened here is that we're keeping ourselves healthy, and so we're living longer. So there's been a huge decrease, for instance, in the age-specific instance of cardiovascular disease. So for a given age, people are healthier. And that probably also explains declining age-specific impact of dementia, because it looks as though cardiovascular disease is a major risk factor for dementia. But because people are living longer, the overall prevalence is going up. Because they're living long enough to get ill despite the age-specific decline. So the major thought in people who work on biology of aging I think now, is that because advancing age is the major risk factor for these many different age-related conditions, not just disease, but frailty and sensory impairment and so on, that perhaps what we should be concentrating on is the aging process itself and thinking about its mechanisms and looking at whether we couldn't intervene before the onset of disease into aspects of mechanisms of aging and prevent multiple different age-related conditions simultaneously. So the aim of the kind of research that I and many other people do is to try to compress that period of ill health and disability at the end of life. It's not to try and make people live longer. That's happening already. It's to try to make the end of life less unhealthy. And I think really the idea that that might be possible has come from work on these creatures, the laboratory model organisms. So budding yeast, the nematode worm, C. elegans, Drosophila, and mice. I do a lot of my work with these two. And of course, they've been incredibly powerful engines for biological discovery, really because of the principle of evolutionary conservation. So for a lot of processes like metabolism, genetics, you can start with a simple organism, even a bacterium, and work out what's going on there, with a very good chance that what you find out will be conserved in these other more complex organisms. You can often take a gene from a fly or even from a human and put it into yeast, and it can replace the function of its ortholog in the other organism very well. There's very strong evolutionary conservation of biological mechanisms. But I think there was some doubt about whether this would be true for aging. First of all, as you've seen, the way it evolves is rather haphazard, doesn't lead strongly to the idea that mechanisms would be conserved. And also these creatures, they live for very different lengths of time from each other. And the kinds of stresses and challenges that they face in their everyday lives are very different. But finally, somebody who's my personal heroine in this whole story did the key experiment. She was working with C. elegans, and what she did was a very simple mutagenesis. She just took the worms, fed them a chemical mutagen, and asked if she could isolate strains of worms that were long-lived. And she found that she could. In fact, they were dramatically long-lived. This was Cynthia Kenyon. So this is one of her mutants. It's called Dept, and this is a survival curve now, and you can see the huge increase in median and maximum lifespan. It's really very substantial. And it took a while to figure out what this gene was, but eventually it turned out to encode the single worm insulin-like growth factor receptor. Actually, it was not suspected that those pathways were present in invertebrates until she did this work. So that was interesting. What the mutation did was to reduce the activity of the insulin receptor, and great interest. As you can see, it was published in Nature. But I think it was also thought probably that this might turn out to be a worm peculiarity. But eventually, the pathway came to light in Drosophila, not because of anything to do with aging, because of its importance in the control of growth and metabolism there. And it turned out that mutant flies also were long-lived. So this is Dept again. This now is a fly that's lost both copies of a gene called Chico, which encodes the single fly insulin receptor substrate. There are 4 of them in mice and in humans. And this is a Chico mouse that's lost both copies of the insulin receptor substrate 1. So you can see there's very strong conservation of the effect on lifespan. Now, of course, we're not really interested in lifespan. What we're interested in is health. And it turned out that in the invertebrates, by any measure of activity, stress resistance, learning and memory in the fly, the mutants improved performance. And this mouse was quite thoroughly phenotyped at the time. And you can see this really very broad spectrum improvement in health in different tissues and different systems that you wouldn't superficially think had much to do with each other. So they had better glucose homeostasis as they went through middle and old age, better immune profile, better rotor rod performance. They got less osteoporosis. You can see the cataract here on the control, and also this ulcerative dermatitis on the head and nape. About 40% of the controls got that at some point during aging, completely absent in the mutant mice. So I think what's interesting about this is the very broad spectrum prevention. So, of course, the important thing here is this conserved in humans? And as soon as it comes to humans, it's harder to make progress. But the main way that people are looking at this at the moment is through genetics. So taking the human orthologs of the genes that have turned out to be important in aging in the animal models, and looking at whether specific mutations are associated with survival to advanced stages into the 90s or 100s, either in sporadic individuals or in whole families. And that's turning out to be quite interesting. And the main one that's turned out to be highly repeatable so far, and also the only one to turn up in a GWAS study, is this FOXO3A gene. So what that gene encodes is a forked-boxo transcription factor. And the reason that it's important is that there's just one present in the genome of the worm and the fly, and if you knock it out, the lifespan of the animal is no longer increased by reduced insulin IGF signaling. So it's one of the key transcription factors that's activated by reduced insulin signaling, goes into the nucleus, alters the expression of genes involved in immunity, stress resistance, oxidative damage, all the various things that go wrong during aging, and protects the animal. And this gene in humans has some specific variants that are associated with survival to advanced stages. There are also studies simply looking at the activity of the insulin pathway, or this TOR pathway that's very closely associated with it. And again, finding that there tends to be lower activity in families that live to advanced stages. So it's looking interesting. I think it's early days with humans, but I think there's cause for optimism that these effects might be conserved. So what we've got here is a signaling network that matches the costly activities of the organism, its growth, its metabolism, its reproduction, to its current nutrient and stress status, and sets the system accordingly. And this turns out to be very important in combating the effects of aging. Now, I've just told you about one of the earlier systems that turns out to be involved in the control of aging, and one that I'm particularly involved with. But as a result of work in many labs looking at different aspects of it, it's turned out that aging does have highly characteristics and very conserved evolutionary mechanisms. They've come to be known as the hallmarks of aging. So it looks as though the whole process kicks off with something going wrong at the genetic level. So either damage or mutation to the genetic material itself, the way that it's packaged in the nucleus, the molecules associated with it, or erosion of telomeres, all mean that patterns of gene expression start to go wrong in cells. And then we start to see very important loss of protein handling. So both protein synthesis and control of degradation pathways goes wrong during aging. I've talked about nutrient sensing. What seems to happen here is that these signaling pathways, which have evolved because of their effect in the young, drive too strongly in the old cells and become damaging, and this is why tamping them down helps. We see a lot of mitochondrial dysfunction, fusion-fission goes wrong, you start to see leakage from the mitochondria, their function generally goes down during aging. Cellular senescence is a relatively new kid on the block, but it's turning out to be very important. So what happens here is that normally during development and during wound healing, senescent cells appear because they're important in tissue basically remodeling. And when they've done their tissue remodeling in those contexts, in a young individual, they're removed by cells of the immune system. So it's a regulated process that's eventually terminated. But as aging progresses, they aren't removed. The immune system fails to do its job. They start to accumulate in tissues, and they have a set of cytokines that they secrete, known as the senescence-associated secretory phenotype, which is extremely damaging in tissues. And it turns out that if you experimentally remove these cells in a mice, either genetically or with a drug, you can both extend its lifespan and improve its tissue function. So this is probably another example of pleiotropy as well as nutrient sensing. These cells are very important earlier on, and they're a very important anti-cancer mechanism, because what they do is to limit the proliferative potential of a damaged cell. But during aging, they become damaging in their own right. There's a lot of problems with stem cells during aging. They either disappear completely or they fail to respond to the normal signals that would cause them to divide and repair and renew the tissue. And there are various forms of altered intercellular communication. So a very important one is of sterile inflammation that tends to increase with aging in all of these organisms. But also communication with the microbiome in the gut goes wrong. There are alterations to the circulating systemic environment. So all of these things go wrong during aging. They're not independent. Often one knocks onto the other, and they can interact with each other. There's communication between different tissues during aging. So it's a complicated process, but we understand who the players are, and there's a lot of interest in intervening in these different aspects of it to try to improve health during aging. And one of the things that we're particularly interested in our own lab is the effects of drugs, whether we can find drugs that will tamp down the activity of the nutrient sensing network. It's well established genetically that its effects are important, but can we intervene pharmacologically and get some beneficial effects? And this has led us both to discover more about how the nutrient sensing network works in aging, as well as come up with some ideas about how we might combat it. And we've been particularly interested in 3 drugs. So one of them is lithium, which is licensed for the treatment of bipolar disorder. I'm not going to say very much about it, but it first became clear in the worm that if you give worms low doses of lithium, it's a very dangerous drug. At high doses, it's extremely toxic. But if you give them low doses, then you can extend their lifespan. That's shown in 3 different studies. And we eventually showed the same thing in Drosophila. So this is a cartoon of this nutrient sensing network in Drosophila. We've got the nuclear membrane here. Here's FOXO, this transcription factor that I mentioned. This is the cell membrane. There are a lot of ligands in the fly. There are 7 different insulin ligands, but the intracellular pathway is much more simple than in mammals. There's just one of everything, whereas mammals usually have 4. And when we go below the receptor here, when it's stimulated by the ligands, this is the insulin receptor substrate. The pathway bifurcates at this point into what's become known as the canonical insulin signaling pathway, because this is what regulates blood glucose. But actually, it also communicates directly with this RAS pathway, which is very well known in a completely different context, because about 1/3 of human tumors contain some sort of RAS mutation. It's very important in cancer, but largely ignored until quite recently in the context of aging. And then there's this intracellular TORC bit of the pathway, which is very important in sensing amino acids, as well as certain types of stresses. And lithium is important because it inhibits this protein called Shaggy in the fly. This is GSK3 in mammals. And we showed in Drosophila that, as in the worm, it extends lifespan. It does it definitely by inhibiting Shaggy. We can show that by its interaction with mutations in Shaggy. And it controls a different transcription factor from this canonical pathway called the Kaplan collar, or NRF2, in Drosophila. So it's definitely geroprotective in the 2 invertebrates, and we're actually looking at what it's doing in mice at the moment. But what I want to do is to talk about these other 2 drugs. The first of them is trametinib. It doesn't actually directly inhibit ERK. It inhibits something that activates ERK. I'll explain in a minute. But it's a drug against the RAS pathway is what's important about it. And I'll also talk a lot about rapamycin, which inhibits TORC1. This is probably the most prominent geroprotective drug that we have at the moment, and is really looking very interesting. But I'll say a bit about trametinib first, because it too is looking quite interesting. So as I say, there's this bifurcation in the pathway here, and because of the discovery of FOXO by Cynthia Kenyon, it was assumed really that this branch of the pathway is what's important in aging. But we decided eventually to just have a look at what's going on here. And this is done by 2 very talented postdocs who've both got their own research groups now. And Kathy was really the geneticist on the project. And what she did was to look at Chico, which is a very highly conserved protein. These insulin receptor substrates evolve slowly. They have very highly evolutionarily conserved structure. And they are the key to the interaction with the 2 pathways, so the canonical pathway down to FOXO and the RAS pathway down to some specific transcription factors in the fly. And what she did was to produce mutant flies where she'd gone for these conserved residues in the Chico protein and made flies where either Chico could only communicate with the RAS pathway or could only communicate with the canonical pathway. So she could break one or the other of these connections. And she could show, looking in cells, that she'd done this successfully. It had already been, these sites had already been worked out by Morris White and Ernst Haffen previously, and they work very nicely in the fly. So what they then did was to look at what these mutant flies did with their lifespans. So what we have here are the control flies in black. This is the Chico positive control, which should be long-lived in red, and it is. These are where she puts back a wild-type Chico into the flies, again, negative control, and you can see the lifespan comes back to the control value. These are the two mutants, and what you can see is that they're both long-lived. If she breaks either the contact with the RAS pathway or with the canonical pathway, she gets a long-lived fly, so it's acting through both. And it turns out that the story here is that these two pathways have other inputs. They don't just communicate with the insulin receptor. So they're integrating upstream information from different sources, signaling between the two pathways, talking to different transcription factors, one called anterior open and another called pointed for the RAS pathway, which bind at very overlapping genomic locations. So this is quite a subtle system whereby the signaling pathway can listen to different inputs and then subtly regulate the way that the different transcription factors interact according with the inputs that it's getting. So that's an interesting bit of biology, but part of the reason that we looked at this is that, of course, because of its involvement in cancer, there are a huge number of drugs developed against the RAS branch of the pathway. We've been far less successful, actually, in getting drugs for the other branch of the pathway. But the one that we chose to look at for RAS is this one, which is called trametinib, and it's an inhibitor of MEK kinase at this point in the RAS pathway, so it's just above ERK. It's a GSK drug, and it's used for the treatment of a particular type and stage of malignant melanoma. And what we wanted to know was whether it would work in flies. And what these blots here show is that it does. So these are data from cells from flies now, rather than from flies themselves. And what they've done is to use different concentrations of the drug with insulin stimulation. And they've looked at whether this ERK, which is the direct target of MEK, has been phosphorylated. Because if MEK's not working properly, then ERK's not going to get phosphorylated when you stimulate upstream. And what you can find is that's true. The drug really inhibits the phosphorylation. Furthermore, it seems to do it reasonably specifically. So these are two other kinases that could potentially be affected by trametinib. And you can see that there's no effect on them at all if they're given the drug. It's only the phospho-ERK that's inhibited here. So amazingly, there's this drug developed against cancer, but these proteins are so conserved that it really interacts very nicely with the fly protein and inhibits it. And if we feed the drug to the flies, it extends their lifespan. So this is shown here for dose-dependent increase. And you can see for these two, the purple and the red intermediate doses, we get a nice increase in lifespan. The blue, we've overdone it. Interestingly, it's very bad for young flies, but it still extends the lifespan at older ages, but too high overall concentration. So we're very encouraged by this, and we're actually starting to look at trametinib in mice now, and we're finding so far that it does indeed extend the lifespan of male mice. And in both organisms, we're really starting to look hard at the mechanisms by which this branch of the pathway is working. But the other drug that I would like to talk about is rapamycin, which, as I've said, is probably the most powerful geroprotector that we've got at the moment. And it disrupts this complex here, which is called TORC1. So it's a protein complex that contains the TOR kinase. And what rapamycin does is disrupt the interaction between a particular pair of proteins, and so it inhibits it. And this complex has many different outputs. It's a really central regulator in the cell, has many different stress and particularly amino acid inputs, and a number of downstream things happening from it. And importantly, it can extend lifespan in flies. So this was shown by Ivana some years ago. She now also has her own lab. And you can see this more or less dose-dependent increase here. And with flies, it's relatively straightforward to ask how a drug is working by simply looking at what it does in a mutant fly. And two very well-known outputs of the TORC complex are, first of all, that it phosphorylates something called S6K, this molecule here. And Ivana could show that if she put in a version of the S6K which couldn't be inhibited, then she no longer saw the extension of lifespan. So that inhibition is important. And it also activates a process called autophagy. This is a cellular cleanup process which goes on at a basal rate in cells. So basically, organelles that have become damaged and need replacing, similarly macromolecules, are recycled through this process called autophagy. And also, if the animal is starved so that it starts to need nutrients, then autophagy is induced as well. So there's a basal and induced level of the process. And under starvation, what it does is to recycle nutrients for really key processes in the cell. And it was rapamycin, which is actually, it's on license use as a drug, is to prevent tissue rejection after transplant, particularly kidneys, to prevent restenosis after cardiac surgery, and also as a cancer chemotherapeutic in several different forms of cancer. And very importantly, it can extend lifespan in mice. So this came out of work led by Richard Miller in the US. They have a program there, the Intervention Testing Program, where each year people can put forward drugs to be tested in mice for their effects on lifespan. And they test them in 3 different centers simultaneously. So it's a very robust test. And you can see here the nice dose-dependent increase in both sexes. It looks weaker in males, but that's because they detoxify the drug more. So for a given concentration in the food, the circulating level in the blood is lower. But it clearly, in both sexes, a nice dose-dependent increase in lifespan. And this has been one of the few drugs so far to go through from this kind of discovery route to humans. So this is work from Joan Manick, who started out in Novartis. She now has her own spin-out company called Restore Bio. And what they've done here is to show, I mean, as in old mice, old people respond very badly to immunization against flu. So they don't mount a good immune response, and they don't show very good protection in the ensuing winter. The immunization works much better in the young. And there was a previous study showing that if you give mice a dose of rapamycin, allow them to clear it, and then immunize them, this brings the level of immune response and resistance to the flu virus in the old mice up to the level of the young mice. And this is more or less an equivalent study. So she took a group of old people who were about to be immunized. They were pre-dosed with rapamycin for 4 weeks, allowed to clear the drug, and then immunized. And she saw both an increase in the immune response and, again, fewer respiratory infections in the ensuing winter. So it looks as though, specifically in the elderly, it really can enhance that type of immune response. So these are interesting drugs, and they're all used clinically, so they have a clear safety profile. The idea is that they could be relatively straightforwardly repurposed against aging. It's not a question of discovering new drugs, which would be extremely difficult for aging. We've got these drugs that are showing the potential to be repurposed. But at the clinical doses that they're generally used, and with the kind of dosing regime that they use, they do have quite serious side effects, all of them. So there's a lot of interest in trying to find out if there are ways that we could reduce the dose given, either just by giving less, possibly by combining drugs, or possibly by intermittent dosing regimes. Could we get the doses down? And one of the things that we've looked at in the fly is this idea that we might be able to combine drugs. Because that signaling network that I showed you is much more complicated than I indicated. There's a great deal of feedback. If you inhibit one node, quite often the others will pop up and try and fight what you've done to bring the system back to normal. So we may well need to inhibit multiple modes simultaneously. And this is something that we've looked at in the flies, and we actually got quite dramatic results. So I've mentioned the 3 drugs that we're looking at: lithium, rapamycin, trametinib. And this is a case where we've combined them. So this was work from Jorge in the lab, he did his PhD at UCL. He's now in Harvard. And what he's done here is the combinatorial experiment. So he's given them lithium, rapamycin, and trametinib on their own at the dose that gives the maximum increase in lifespan. He's then done the 3 pairwise combinations, and finally he's combined all 3. And you can see that you get a nice response with each of them. The doubles are much longer lived, and here's the triple And actually, these drugs add completely additively to each other. There's no interaction, positive or negative, between them. They simply add on in each other's effects. And this is now a 43% increase in the lifespan of the flies, by far the largest that we've ever seen with anything. So it looks as though this kind of combinatorial treatment might be a particularly powerful way to go. But the other one that we're very interested in is intermittent dosing, because it's turning out that, at least for rapamycin, there's quite an extraordinary memory effect of past treatment with the drug. So this was the experiment that we did where we first saw this. It was done by Paula, who's a postdoc in the lab in Cologne. And what she's done here is the standard experiment that I've shown you. Give the flies rapamycin, and you find that with chronic treatment it extends their lifespan. But what's included here is this red group, and they were only given the drug for the first 2 days of adulthood. The drug's then withdrawn. We can show that talk activity goes back completely to normal. There's nothing that you can see in the signaling domain. But you then just leave the flies and wait till they start dying. And they live just as long as flies that were treated continuously throughout life. So there's this extraordinary memory of the early treatment, and we can't see it at all in the signaling domain. It's something else that's been locked down. So obviously we wanted to know, what's the mechanism here? And one of the big hints of at least where to look for what might be going on comes from this, which is the sex difference in the response to rapamycin. This is Jenny Reagan, who has her own lab in Edinburgh now. She was a postdoc in the lab. And what she's done here is a lifespan experiment with males in blue and females in red. And you can see the males just don't respond to the drug at the same dose. And no matter how much rapamycin she gives them, she cannot extend the lifespan of males. So there's a huge sex difference here. And in flies, not in mice, but in flies, this is a very strong hint that you're probably looking at something to do with the gut, because the gut ages very differently in the two sexes. This is shown here. The fly gut is not so dissimilar to a mammalian gut. It's very highly regionalized. Different bits of it look different. And many of the main bits consist of a columnar epithelium with stem cells at the base of it. So these long columnar cells that line the lumen of the gut. And what we've got here are females at the top and males at the bottom. And here you can see this nice columnar epithelium in the male. So this is a section through it. So the gut itself is here. And you can see these nice epithelial cells all beautifully lined up with their blue nuclei. And as the fly gets older, going to the right, nothing happens. He's just fine, even to very old age. This is looking down on the surface of the cells. So you're seeing the tips of these long columnar cells. And again, you can see that the youthful arrangement stays present throughout life. So male guts are fine during aging. Females are not. So if we look at the same thing here, they start out fine. But even in a relatively young fly, so she's fully fertile at this age, you can see that things are starting to fall apart. The nuclei aren't so well lined up. You start to see misdifferentiation of the stem cells. They start to form little misdifferentiated clusters, sometimes called tumors. You start to see wound healing rosettes, and eventually, by the time she's old, the gut is basically a complete mess. And you can see the same thing looking down. Here's a clear wound healing rosette. So what happens is there's considerable gut pathology in an aging female fly. And it turns out that rapamycin completely rescues that pathology. So if we look at two manifestations of it here, these are just pictures of the gut here where you can see the disordered appearance in an old female here. This is with chronic rapamycin treatment. This is with past rapamycin treatment. So this is looking in day 50 at flies that were actually treated up to day 30 this time. These are tumors, these misdifferentiated stem cells that you can see in the gut. And you can see that both chronic and past rapamycin treatment drop them right down, and to an equivalent extent. And as a result of the visible pathology in the gut, the gut actually becomes leaky as the female gets older. It no longer keeps its contents in. And in flies, you can measure that with this blue food dye. So in a normal, healthy fly with an intact gut, the dye stays in the gut, which you can just see here. If it's leaky, you get a blue fly. And you can see again here that both chronic and past rapamycin treatment knock this right down. There's almost no gut leakiness as the female ages. So the drug rescues it. So the question is, how is it doing it? And I mentioned that one of the important outputs of inhibition of TOR by rapamycin is you see this increase in autophagy, this cellular cleanup process. And it turns out that that is a very important player in what's going on. So here, what we've got is one measure, we've used many different ones, of induced autophagy in the gut. So this is a control female. This is one that's chronically treated with rapamycin 12 days after she's taken off the rapamycin. And you can, here, and you can see the nice increase in autophagy in this red marker. So that's marking things called lysozymes, which are an indicator of increased autophagy. You can see that even 12 days after the fly's off the drug, she's still showing this increase. It's quantified here. And there's no difference between the chronic and the past treatment. So autophagy is coming up with rapamycin treatment, and it's staying up after the drug has gone, and this is associated with this phenotype here. So what happens in the fly gut as it ages is that as those big columnar enterocytes start to become damaged, this usually takes about 2 weeks for each cell. When the cell's about to die, it sends a message to the stem cells to be renewed, and the stem cell divides and differentiates into a new enterocyte with a turnover rate, as I say, of about 2 weeks under normal conditions. And this is measuring turnover rates here. And you can see that with chronic rapamycin treatment, it's way down. This green marker tells us that since we induced it, that cell has been turned over. So here, you can see that 12 days after treatment, almost every cell has already been turned over. But with chronic rapamycin treatment, it's way down, and it stays down with memory. And it turns out, if you actually look into the details of what's going on here, that the enterocytes aren't sending out the signal. They're not asking to be renewed. And so the stem cells aren't proliferating. The proliferation rate goes way down, and you see this lowered turnover rate. So it looks as though the health of the enterocytes is being increased. They're not asking the stem cells to replace them. And, of course, this is all just correlation. What we want to know is if it's causal. And the nice thing about flies is that you can ask that experimentally, because you can manipulate autophagy directly, genetically. And so that's what we've done here. So we're looking in enterocytes now, and what we're doing is directly manipulating the autophagy process. We're increasing it. And you can do that in a cell-specific way in the fly by using this so-called gene switch driver. So this is a particular molecular construct which you can put into the genome of the fly, but it only becomes active if you feed the fly another drug called RU486, so it's inducible. So you can wait till the fly becomes adult and then give it the drug. And it comes on in the it causes the construct to come on in the enterocytes, and it can then drive another gene. And what it's driving here is this gene called ATG1. And ATG1 is a master regulator of autophagy. And if you increase its expression in the fly, you increase the autophagy process. And you can show that molecularly. So what Paula's done here is to overexpress ATG1, or give the flies rapamycin, or give them both. And you can see that rapamycin and ATG1 both increase the lifespan of the fly. This is the chronic situation here now, so both are permanently on. And if you put the two treatments together, you don't see any further increase in lifespan. So it looks as though the autophagy is what is accounting for the effect of rapamycin. Here she's now done both things inducibly. So they've only had autophagy turned on for the first 2 weeks of life. They've only had rapamycin for the 2 weeks of life. And what she finds is exactly the same result, that either way you can increase the lifespan of the fly, but if you put the two together, they don't live any longer. And what this is telling us with various other measurements as well is that if you induce autophagy here, it actually stays up for the rest of the life of the fly in these cells. Autophagy remembers itself, and that is how this process is working. And it turns out that that's a result of a positive feedback loop. There's a molecule that's generated by autophagy. It's called spermidine. And spermidine itself is a very strong inducer of autophagy. So once the process is up, it stays up because it's feeding back on itself. And that's how it's working. So we were, of course, fascinated by that, but these are just flies. We would like to know if the same thing is happening in mice. And that's something that we've looked at. So we've done various experiments with mice, and this has been one of our main experimental designs. So everything, of course, takes much longer in a mouse, but what you can do is to have them born and mature to 3 months. They're fully adult and fully grown by then. And then either they're treated as controls, or they're given the drug up to 12 months of age, or they're given just 3 months of treatment, and then taken off the drug for 6 months. So by the time we look at them at 12 months, they haven't seen the drug for 6 months, and they just had 3 months treatment earlier on. So very similar to what we did with the flies, but taking much longer. And because of the pointer from the flies that the gut might be important, that's what we chose to focus on initially in the mice. And what we've looked at is these Paneth cells, or part of what we've looked at is these Paneth cells. So in the small intestine of the mouse, as in humans, what you have is a very convoluted arrangement of an epithelium that's otherwise rather like the one that I showed you in the fly. And at the bottom of these crypts, so going away from the gut lumen, what you have are the stem cells, as in the fly, that gradually renew the epithelium as the cells become worn out and die. But you've also got these Paneth cells, and they're extremely important functionally in the mammalian gut. They're very important in their own right because they contain granules that contain antibacterial peptides and other enzymes that are very important in gut function. But they also act as the niche, the environment, the key environment for the stem cells. No Paneth cells, no stem cells, they can't survive. They look after the stem cells in the gut. So they perform these very important functions. And also a hint for us is that autophagy is very important for their function. It's known that if you impair autophagy in Paneth cells, they start to show various pathologies. And Paneth cell pathology can be improved by rapamycin. This was shown by the Sabatini group some time ago. So they were an obvious place for us to look at. And it turns out that there is indeed a most remarkable memory of past rapamycin treatment in these Paneth cells. So this is our experiment, just shown again here. And these now are electron micrographs of the Paneth cells. And they contain these dark granules, which contain the antimicrobial peptides and the other things that are secreted by Paneth cells. And this is what they look like in a normal young mouse. So you get these nice, round, dense, black granules. What happens during aging is they develop this hyperdense phenotype. So they get these white circles around the granules, which themselves become smaller. And in a really old mouse, the granules just disappear. And this is probably partly as a result of this process, they become crinophagic, which actually means they're being attacked by autophagosomes and destroyed within the cell. So there's a gradual increase in granule pathology in Paneth cells as the mouse ages. And it turns out that all of these phenotypes are reversed by rapamycin, and they're remembered 6 months after the drugs are withdrawn. There's a really, really clear memory. And the epithelial cells in the gut are held together by these tight junctions, which you can see here in the electron micrograph. And they also become compromised during aging. They become thin and not very electron dense. And you also start to see bulges in them where the two membranes that should abut have started to come apart. And you can score that as pathology. And again, rescued by rapamycin, and it's remembered 6 months after the drug is withdrawn. And as in flies, the mouse gut becomes leaky during aging. You can measure this with this lipopolysaccharide binding protein in the plasma of the mice. And again, it increases during aging, it's rescued by rapamycin, and it's just as rescued 6 months after the drug is withdrawn. So again, there's an extraordinary memory of the drug. And just very recently, we've actually been looking at organoids from these mice. So what you can do is to take crypts from the mice and actually culture them in vitro and look at how well they proliferate and create new crypts and new stem cells. And if you do that, it turns out you get far more organoids from a mouse gut that's had a past treatment with rapamycin. This is a slightly different timescale because we wanted to look at really old mice here to look at the memory effect of the drug. And these are organoids from a control. You can see that they're there, but there aren't very many of them. The chronic rapamycin treatment don't proliferate very well either because they've still got the drug in them. You have to take them immediately from the live mouse. So they haven't yet cleared the drug. But these are the ones that have had the history of rapamycin treatment. And you get an amazingly much larger number of organoids from the gut. This is shown here. And also, if you then reculture from these organoids, you get more crypts and more Paneth cells. So the gut is in much better regenerative condition if it's had a past treatment with rapamycin. So I think what this is telling us is that probably very intermittent dosing with this drug is going to be enough to get a lot of the protective effects. And I think it's also telling us something about the gut, which has been a very much ignored organ in the context of aging, but I suspect that it plays a really key role in many of the things that go wrong. So just very briefly to finish up, I mean, these are the sorts of things that we're interested in our lab, but there are a number of other emerging opportunities from other labs in the field. So I've mentioned cellular senescence, which is turning out to be extremely important in aging, but also extremely targetable. So it plays a key role in a number of age-related diseases, including osteoarthritis. And this is a very nice study showing that if you remove senescent cells from an osteoarthritic joint in a mouse, you get a huge improvement in joint function and a reduction in inflammation. And this, of course, is a protected environment in that it's relatively sealed off from the rest of the body, so particularly targeting joints, but also the eye, where cellular senescence is very important in aging, maybe something that's really coming quite quickly. These are just some of the papers that have come out here. These are the ones showing that in the invertebrate disc regeneration, this was actually a human pulmonary fibrosis study showing that some of these senolytic drugs that kill the cells are well tolerated. These are mouse studies, this one showing that you can extend health and lifespan in a mouse with a senolytic drug. And this is cognitive decline in glial cells, again in the mouse. So I think that area is moving really fast, and we're likely to see some action. I mentioned the systemic environment, although I haven't said very much about it. The importance of this actually came originally from these Frankenstein studies, really, where you conjoin the blood supply of mice. And as long as it's an inbred strain of mice, that's fine. It's well tolerated. You can join them to each other. And you can join mice of different ages. Saul Villeda was really the key here. He can do these very tricky experiments. And of course, you can do this with mice of different ages. So you can have a young control and an old control, but these two are different ages. And the big finding here is that if an old mouse is hitched up to a young mouse, you see a major regeneration of the stem cells in several different tissues, in muscle, kidney, in the neural stem cells as well, and also an improvement in synaptic function in the brain. These experiments aren't particularly practical. They're not ethically terribly desirable, and they're also a bit confounded because if you actually look at films of these mice, I mean, what you've got is a young, energetic mouse who wants to tear around the cage, attached to an old mouse who just wants to go to sleep. So it's a slightly odd situation, but increasingly now they're using plasma and doing the experiments that way, and of course trying to identify the molecules that are important. Is there something bad happening in old mice, or is there something rejuvenating in young mice? Can we either get rid of it or add it? Those kind of experiments. And again, a lot of very interesting and increasingly clinical papers starting to come out on whether this can be applied. The other thing that I haven't said anything about at all, which I think is a really coming area, is the gut microbiome. So, of course, there are huge numbers of commensal fungi, bacteria, and other microorganisms in the gut, in mammals, and it's very clear that they change during aging and also in response to some of these geroprotective interventions. And there have been a couple of very nice experimental studies recently. This is a new model organism for aging research, turquoise killifish. It's come in because it's a vertebrate that only lives 3 months, so about the same as a fly. And this was a lovely experiment for the Valenzano lab, where they simply asked, if we give an old fish a young microbiome, can we improve its aging? And the answer was yes. This was the key group here. These were the old fish that got the young microbiome. These were just various controls. And what they found was that they lived longer, So they accepted the microbiome profile became youthful, and they lived longer, and also they swam better. They maintained their swimming performance. This was a mouse experiment where they took dietarily restricted and control mice. Dietary restriction is the most powerful intervention that we have to protect against the effects of aging. So even in rhesus monkeys, it produces a very broad spectrum improvement in health. And here what they've done is to transfer the microbiome from either calorie-restricted or ad lib-fed mice into a sterile mouse. And what they found was that compared with the control, the dietarily restricted microbiome produced a lot of very beneficial metabolic phenotypes. So browning of the fat, which is typical during calorie restriction, much healthier liver profile, and much less inflammation. So it looks, these are really early experimental studies. There's been an awful lot of descriptive work on the microbiome, but I think increasingly these kinds of experiments and figuring out which bacterial taxa are important, what metabolites they're producing and so on, is going to be very interesting. So I put Edith Saunders in. I hope she would have approved of this stuff. And what we can conclude is that aging evolves as a side effect, as we've seen in mutation pressure and selection at younger ages, but it does have conserved mechanisms, and we can intervene in them in various ways. And I think what we're really hoping is that this is going to lead to various interventions that can keep people healthier in old age. And I'm going to stop there and thank you very much for your attention.